Hello, I'm Alice Williamson, and I'm a PhD student at the Institute of Metabolic Science at the University of Cambridge. I'm going to share how we leverage genetics to investigate the role of melanocortin 3 receptor, MC3R, in human physiology. The state of somatic energy stores is communicated to the brain, which subsequently regulates key aspects of behavior, growth, nutrient partitioning, and development. The central melanocortin system is one such pathway, which regulates the control of appetite, food intake, and energy expenditure through MC4R. However, the exact role of MC3R, which is the only other melanocortin receptor that is expressed in the brain, has not been determined to date. Examining whole exome sequencing data in UK Biobank, we identified that individuals who carry a predicted damaging MC3R variant have a delay in puberty, shorter height in childhood and adulthood, lower IGF-1 levels, and reduced lean mass. We identified variants covered on the genotyping array in all 500,000 individuals and functionally characterized these in vitro to identify a dose-dependent relationship with variants identified to be complete loss of function having the largest effect on phenotype. We saw this pattern association for puberty timing, but also for reduced height and lean mass. All the people identified in UK Biobank were heterozygous for the MC3R mutations. Despite it being rare for individuals to carry 2 copies of a deleterious mutation, we identified an individual in the Genes and Health cohort who was homozygous for a complete loss of function MC3R variant, meaning in effect he has no functioning MC3R. This individual had a more extreme phenotype to that of the carriers of the heterozygous variants in UK Biobank. For example, he had a significant delay in puberty with onset in his early 20s. Using DEXA data from the proband and also UK Biobank, we were able to compare his body composition to that of ethnically, age, and BMI matched controls, showing that he has markedly lower lean mass compared to all 3 groups. Following our studies in humans, we identified in mice that the expression of MC3R is enriched in hypothalamic neurons controlling reproduction and growth. In combination, these findings suggest that the central melanocortin pathway integrates nutritional status to control through MC4R the acquisition and retention of calories, while MC3R primarily regulates their disposition into growth, lean mass, and the timing of sexual maturation. This work has been a huge collaborative effort, and I would like to thank all groups involved in this research, as well as the participants of the UK Biobank and Genes and Health cohorts. Thank you for listening.